As the cost of next-generation sequencing, or NGS, continues to decline, it has become a routine part of research for many laboratories. Generating the sequence data is only the first part of using NGS in biological research. To turn this data into insightful information and to answer biological questions, complex bioinformatic analysis is required. Bioinformatics is an interdisciplinary field that bridges biology, computer science, mathematics, and statistics. A major focus of the field is addressing the complicated analysis of NGS data. The bioinformatic analysis of NGS data can include many steps, with multiple software tools used in each step, such as quality control assessment steps, sequence alignment, data summarization into genes or regions, data annotation to genomic features, statistical comparisons, or multi-omic integration. With so many steps, a single analysis can easily become a tangled web of code, with challenges to portability, reproducibility, and maintainability. However, this won't be the case for much longer following recent developments in workflow management systems such as Nextflow language, turning this complicated mess of code into automated systems. This, in combination with recent developments in software, will enable portable, reproducible, and community-maintained bioinformatic workflows. These new technologies will lower the cost and complexity of bioinformatic analysis by making it easier for laboratories and cores to perform routine bioinformatic analysis, and for bioinformaticians to publish and distribute best practice workflows that run in a variety of environments.